everyone, welcome to today's MPG Primer. Today we have a very famous MPGer, Kaitlin Samocha. She is an assistant professor in the Center for Genomic Medicine at Massachusetts General Hospital and an associated scientist at the Broad Institute. She received her PhD in genetics and genomics in 2016 from Harvard University and trained as a postdoctoral fellow at the Wellcome Sanger Institute. The focus of her research career has been on developing methods and statistical tools to improve interpretation of genetic variation, particularly rare variation. And today she'll be giving a talk on modeling rare variation and calculating genetic constraint.
Okay, good morning. Thank you very much for that intro. By famous, it means I was recruited to lead the seminar after this, so hopefully I'll see some of you all there. So this is one of the MPG primers that's supposed to be part of these high-level intros to topics. If you are an expert in this, a lot of this is going to be review, and we are going to touch on many things at a high level so that you have a flavor of what's going on. I'll try to highlight particular publications that I think are good to read or check out if you'd like more information, and I'm obviously happy to provide more information later. So today we're going to talk a little bit about mutations and mutation rates and why we think that's an important and a cool thing to look at. I'm obviously extremely biased. I think quite a lot about these. We will also be looking at how we can then model rare variation in the human genome. The focus here is going to be on coding variation, but of course, as more genomes become available, non-coding variation is going to be included as well. And then how we can use those mutational models in two primary ways. The first is to identify genes that appear to be intolerant to mutations in the general population. These are constrained. And the second is to empower gene discovery, specific in a rare disease setting. So first off, as the highest level reminder I can possibly give you, we have a lot of genetic variation within our genomes, and the scale of this genetic variation is tiny to massive. So starting at the bottom here, we have single nucleotide insertions, deletions, and single base substitutions. So a lot of people refer to these single nucleotide changes as single nucleotide polymorphisms or SNPs. I tend to call them SNVs or single nucleotide variants themselves. Polymorphisms to me has some sort of frequency information, but there will be people who will fight me on that, and that's fine. A little bit larger, we have microsatellites, and as we move up the scale, we have mini satellites, these retrotransposon insertions like line elements, allos. We then get to tandem repeats, so duplications, deletions, inversions, and then all the way up, of course, to aneuploidy. And this is just to remind you that there's a lot going on in the genome that we have to keep in the back of our mind at any time. Speaking of a lot in any individual person, we're talking about 4 to 5 million genetic variants. So this is displayed quite nicely from a paper from the 1000 Genomes Project. This is from 2015, but we see similar patterns now. And you can see across these different population groups that are colored similarly if they're closer to each other in terms of global ancestral relationships. You see we rise from about 4 million in the European-based populations up to about 5 million in the African-based populations. Just really want to emphasize here, this is not including some of the advances in structural variation that we've been able to call recently. Some of the telomere to telomere and the long reads have allowed us to pull out more complex structural variants. So it's an even more complicated picture than this. This still is getting you after the fact that there's a lot in every individual's genome. So we like to come up with ways to prioritize what might be important. And this is just a summary slide so that we're all using the same terminology here. So in particular, I just want to highlight that this last class of variants here, nonsense, frameshift, and splice site, I will try to refer to collectively as PLOF, or putative or predicted loss of function variants. There's also PTVs or likely gene disrupting. I will try to use PLOF here. And the reason we don't just use loss of function is because not all of these are real loss of function variants. That's just a nice paper on that. And just as an emphasis, synonymous or protein unchanged, missense the protein has changed, and this is a category that's a real mixed bag. Some of them seem to be benign, fairly neutral, and some of them seem to be quite deleterious. So this is a genetics crowd, so you all look like you're on board, and we'll swiftly move onwards. So speaking of that 4 to 5 million genetic variants in every individual's genome, we are talking about 10 to 12,000 synonymous variants and up to 150 PLOF variants in this. including some disease-reported variants that you can see in databases like ClinVar. So some of these are expected to be seen in general populations if they cause an adult onset condition. Maybe someone has made it into the study before the condition has started. However, some of them are going to be incomplete penetrance. And I know that Heidi will eventually be presenting on some work about interpreting variants and what it means when we say potentially pathogenic. But we can take all of these variants and try to focus just on the rare ones. So here we're using a population reference database called GNOMAD, or the Genome Aggregation Database, to filter to the number of rare variants we see in individuals' exomes and genomes here. And across the board, you can, across populations, again, colored by the different genetic ancestry groups, we see that it's about 10 to 20 loss of function variants, 100 to 200 missense or small insertions and deletions, and a little bit less for synonymous variants. So even if you're looking at the rare variants, less than 0.1%, we're still talking about quite a lot going on. But these reference populations are really helpful for us, and they empower some of the studies we're going to talk about a little bit later. So when it comes to the mechanisms by which mutations arise, that is a totally separate multi-hour lecture that someone gives in an undergraduate class. We're not going to go into all the depths here. I just want to highlight that Jonathan Pritchard's recent textbook on the studies put in for free online had highlighted mutations in 3 general contexts. So germline, somatic, and cancer. And I thought that was a nice breakdown. So you could argue that cancer is sort of a subset of somatic, and I would say that's true, but there's different properties, there's different mutational spectra, and there's different rates that we see in these contexts, and that's why it's nice to split out. Today, we're focusing on the germline. So that means that we're talking about what, when one was made as a zygote, all the passages down to the male and female primordial germ cells, and then as they develop to the end stage germ cells, the sperm and the oocytes. So there's a lot of different opportunities here for mutations to arise, and it's mostly when the DNA is replicating through all of these different cycles. So DNA replication is extremely good. Like, it's a very error-less mechanism. It does not create mutations that frequently, but we have 3 billion base pairs in all of our cells, and every single replication time point, it all has to be copied over again. So even something with a really high ability to, okay, I'm seeing a lot of Q&A. Even something with a really good ability to replicate without errors, we are going to still see a couple pop up. And when we look at de novo mutations, so those that are newly arising in the child, so you didn't inherit from one of your 2 parents, we do see fairly consistent differences between different mutational classes. And we'll get a little bit more into this, but there's certain mutational spectra that are associated with sort of replication itself and with CPG mutations, which we'll talk about more. And that's the major driver of a lot of the signatures that we see in the germline. So there have been a lot of approaches over the last decades to try to understand the mutation rate broadly. In the past, we had to do indirect ways of estimating this, which was looking at, let's say, humans and chimps. So it was divergence of 2 species. And this required that not only did we have some sense of the number of differences between the 2, we also had a sense of the distance between the 2 in the genealogical tree. Those estimates have now been replaced a little bit, or at least are being compared to a more direct estimation. And this is, again, when you're sequencing a parent-child trio. So we can directly sequence to see all of the mutations that are new in the child that were not seen in the parents at all. So over the years, we've sort of settled on this 1.2 times 10 to the minus 8. That's the kind of key number to remember. This is our base substitution rate across the genome in humans. If you look at this, and this is, again, I took this from Jonathan Pritchard's new textbook because I liked it to put it all in one place. If you look across the genome, we do see that this varies between different types of mutations. So small insertions and deletions are actually much, much more rare than single nucleotide changes across the genome in terms of their mutation rate. And we see even more rare for these large structural changes with, of course, the reminder that they impact more bases, and therefore they're potentially a bigger hit.
Hey, Kaitlin, can I ask you a quick question? Yeah. I know these are all measured from live births, but are there any studies to look at pregnancies that don't result in a live birth that maybe it's actually a little bit higher mutation rate?
That's a great question. So there are some consortia that have started now that are trying to sequence fetal loss. That obviously has a number of ethical and regulatory hurdles to get through. I don't recall if they were seeing noticeably higher mutation rates. It might be a little bit higher, but that could also be ascertainment, of course. The hypermutability leading to a lot of de novo mutations, if one of them lands in a sort of negative or a sort of a deleterious gene, then you're going to be ascertained into one of these cohorts. This is a fairly stable number. There's some work from Kelly Harris's lab about potentially slight differences between population groups around the world. So I am oversimplifying. So that's a good point. Yes.
Kaitlin, kind of a related question that came through the Q&A is, how do you really think differently about a somatic mutation versus a germline de novo? And, obviously the de novos had to arise in some cell that was probably part of the gamete, but I guess, yeah.
Great question. Okay, so this is about somatic versus gamete being different. It's mostly, when I'm talking about somatic, I mean in the context of an adult human tissue that's gone through many more passages. Yes, of course, a de novo has arisen in some cell type at some point, but there are certain processes that appear to have a larger impact in the collection of somatic mutations across a time point than they do in the germline. An example is transcription coupled repair. So this is where the transcribed strands of DNA, and I was going to be on a slide later, the transcribed strand tends to be corrected with higher fidelity than the non-transcribed strand. And you see that much more noticeably if you're looking at somatic tissues than if you look at the germline, though we do see that signal in the germline as well. That's one example of how they might be a little different. I would also say mutagenic agents are more likely to influence somatically arising mutations versus what's happening in the germline. So there was a paper recently about some cancer, what was it? A drug given for a certain kind of cancer that also influenced the de novo rate, particularly from fathers. So there's potentially some mutagens that can influence your germline mutation rate as well. And obviously we care quite a lot about knowing those because we don't want to give people these kind of medications and then tell them that it's before they have had children or before they've had all the children they want to have. Right, so these overall mutation rates, they're great. They're actually really, this is intro. We really want to get to a way to figure out for any given stretch of DNA, let's say a gene, some collection of DNA that we care about, the mutation rate for that or the mutability of that. And the reason that we want this is because it allows us to do a lot of more specific follow-up. Today, we're going to focus on two. So the first is selective constraint, or quantifying the amount that selection has depleted variation within the human population. And the second is gene discovery. I just took CDK13, but a lot of times when we're looking at rare disorders, we're looking at de novo mutations, and we need a mutation rate in the background for that. I should note that there are obviously lots of other applications of having a gene-specific mutation rate. There are demographic models and history that you can understand by using this information. So these are just the two we're focusing on, but there's quite a lot. And I just want to really emphasize that this is the motivation here. These can be extremely useful. Oh, okay, I didn't skip that. Great. Let's talk about modeling rare variation itself. So what factors could influence mutation rate? Someone wants to shout it out. Anything that they think makes sense. I have an order here, obviously, but I'm happy to chat. No, too early? Too early. I already told you about one of them, base composition, right? When we look at de novo mutations in a child coming from unaffected parents, we see, for example, that there's a lot more C to T than there is C to A. So clearly, the base that is being mutated from and to matters. However, we've also seen that local sequence context can have a big impact. I'll note on this plot here, they've actually explicitly separated out CPG to TPG. So this is because when you have a C right next to a G, this CPG dinucleotide, what happens is that cytosine can be methylated to make this 5-methylcytosine that's very easily deaminated to thymine. So normally, cytosine to thymine without this methylation is a super rare event that doesn't really occur as highly as this methylation to deamination. So this is the most common single base change across the genome, and it's this methylation signature. But that means you have to have methylation there. That also means you really want to know, not only do you have a C as the reference that you're looking at, you want to know what's right next to it, but how much local context should, this one, how much local context should you take into account? So obviously we want the bases right to the left and the right. Clearly I've put an example with a C next to all these Gs. So clearly this would be a site that if it was methylated would be highly mutable. So in the past, we've had a lot of publications locally focused on using this trimer rate. So this is looking at the trinucleotide with the base in the middle being the one that mutates. So we can predict GCG becoming GAG, GGG, and GTG, right? All of those possible single base substitutions in the middle based on just looking at the one base to the left and one base to the right. Now, this is the basis for the mutational models we use in EXAC and NOMAD, and so we're going to go into more detail on this one, and it's just because it's the one that I know the most about, so I will admit bias here. However, as soon as kind of we focused on this trimer rate, there have been other publications looking at, for example, heptamer. So the first was in 2016 from Ben Voigt's group. The heptamer seemed to perform a little bit better. It's slightly more accurate. It captured something else beyond the CPGs that was influencing this local mutation rate. However, there are only, 96 of these, 92, something like that, early, so my math skills are challenged. There's a lot more of these heptamers, and so as you start to look across the genome, this becomes more sparse. It's part of the reason why it's expanding out to 100 base pairs on either side, which maybe would be ideal. There aren't going to be that many representations of this around the genome, just as we expand out. So I should highlight Roulette is a mutational model that Shamil presented on an MPG last year. There's a preprint right now. So they use a pentamer, so 2 bases on either side. And then they actually include up to an additional 8 bases flanking that in a regression model. So this is a much more complicated one than the one we'll walk through. But just to say that people have done everything from 1 base to kind of 12 bases around the mutating site as a way to capture local influences on mutation rate. But there's broader than local influences. 1,000 Genomes observed this quite a while ago, that the SNP density around the genome is not uniform. It's not random, but it's not uniform. So there's specific parts of the genome where we see a lot of SNPs. So everyone's favorite chromosome 6 MHC region, that's, as a non-immunologist, that's just a hard region to deal with. We anticipate there's going to be a lot of genetic diversity there. But you also see the P arm of chromosome 8, chromosome 16 showing up here, both the P and the Q arms. So there's there's a concentration of SNPs here, and that's indicating that something about the mutation rates in these areas, about the mutational processes, is potentially different. And we can guess at what some of those things might be. So for example, replication timing. Replication tends to happen at particular spots around the genome. So the replication fork opens the DNA up, kind of unspirals it, and you start to get the leading and lagging strands being synthesized and separating from each other, and then you have the 2 replications. But it's been found for a while that the closer you are to the replication fork, if you're early replicating, you have a lower mutation rate. I believe the theory behind that is tRNAs and them running out of, like they're getting exhausted or something, but the trend has been established. So if you're here, you're less likely to be mutated than if you're way over here, early versus late replicating sequences. There's also strand asymmetries. So I mentioned before transcription coupled repair. This is one way that you could see asymmetries between the strands. So the strand that is being transcribed versus the one that's not. Leading and lagging strands also, in terms of replication, have different polymerases that contribute, and every polymerase has a slightly different mutation rate itself. So, and a different mutational spectrum. So all of this kind of contributes. Early work in exomes didn't see as much of this, but as we move to genomes, this is a big part. Beyond that, the recombination rate itself, so where recombination hotspots are, also tend to be mutagenic, because you have these strand breakages around them. There are epigenetic marks, particularly which is H3K9 histone marks. Those are associated also with a different mutagenic profile. So there's a lot that we could take into account here when we're considering the overall mutation rate. And a lot of these have been included in recent models. So I should note that the roulette model that I mentioned a moment ago that has that heptamer plus larger sequence does have some 50 kb window for sort of like SNP, or I should say like mutation rate in a larger 50 kb window. They include strand asymmetries. Another model, the Carlson one, which was using the heptamer. One of the heptamer models also had like a bunch of recombination rate epigenetic marks, things all kind of thrown into it. So people are considering these as well. A few other factors you might want to take into account. There's a couple weird regions of the genome, and they're great. I like them, but they're weird. So for example, the P arm of chromosome 16 and the P arm of chromosome 8, which are familiar to us from the last slide, we see clustering of maternally derived de novo mutations. So almost all de novo mutations that we see in people are coming from the paternal line. That's because sperm keep replicating where oocyte kind of freezes in time. You do get some from mom, that happens, but a higher rate are coming from dad. But these are specifically coming from mom, and they have a different mutational profile than the rest of the de novo mutations. So clearly something interesting is happening here. And just to highlight that, yeah, maybe it's some of those favorite regions again that are causing problems, but this is something you might want to take into account. Clearly, there's some interesting mutational processing happening in these particular spots. And then lastly, if you're actually going to use these mutation rates to try to predict what you'd see in a realistic cohort, there's like theory, like how likely is it to mutate versus practice, like I have some data in front of me, you might also want to take into account how well we've sequenced that region. This is particularly important for exomes. So I've hacked a MEF2C picture from the Nomad browser just to say that exome coverage is these light blue peaks, and you can see some exons are super well covered. However, some exons are not. Genome coverage is a little bit more stable at 30x, but there are still parts of the genome that we do not sequence particularly well. And so if you're trying to use these mutation rates in real data, you might want to take into account that some of them, certain places, you can predict, like here, you can predict mutations there, but you might not see them. And you don't want to count that against yourself. So this is a correction that I'm a little biased for because I think it's important anytime you're doing something beyond theory. Great. So we're going to talk about Nomad's mutational model because I'm biased and because I think it's one of the more straightforward ones that we've used for a lot of our constraint scores. So we're using this trinucleotide context, right? One base to the left, one base to the right. We started by looking at SNPs, so these single nucleotide changes in intergenic regions. So these are in between genes. We believe that they're probably neutral in terms of selection. And we were looking at least in the past, ones that were orthologous to chimps. So we had some sense what was the ancestral versus the derived allele. So we know which way the arrow was moving. So we took all of these and we created a mutation rate table. So for every 2 trinucleotide mutating to every other trinucleotide, we had a relative probability of it occurring. We then use that overall mutation rate, that 1.2 times 10 to the minus 8, to move them from these relative probabilities into, oops, hitting my own stuff. Oops, I'm going forward. It's the morning. Amazing. We can then take these relative mutation rates and apply them to the sequence. So you can do a sliding window along it. So for every single trinucleotide, we predict the likelihood of all these other trinucleotides, and we can sum per gene or per exon or per whatever you want the mutation rates by different consequences. So here I've broken it down into the coding consequences because until very recently, almost all of our data was exomes, and so we were focused on coding regions. But in non-coding regions, you can add them up by C to A mutations or C to T mutations. I should just say generally, this talk will not cover as much the non-coding because we are still new there. We have thoughts, we have some early stages, but we are still quite new there. So we additionally, beyond just this local sequence context, which captures those CPGs and captures how big a gene is, right? It just adds everything up. We also correct for methylation. So specifically, we're stratifying just into high, medium, and low methylation bins, which, yeah, baby steps into these places, but we do see that those CPGs that are highly methylated up here have a much, much, much higher mutation rate than CPGs that are lowly methylated. So this mutation rate on the x-axis is from one of our earliest studies, and the one on the y is from the Nomad paper itself. So you can see that kind of before we collapse them all into one category, and adding methylation is helpful. And then of course, correction for the coverage per base. So this was critical when we were making the constraint scores that we'll talk about. Just again, to highlight that if regions had a median coverage less than 10, we weren't really seeing that many mutations there. So if we were expecting them, then we were working against ourselves a little bit. So we can evaluate this in a number of different ways. The way that we've evaluated in the past, and many have, is by looking at rare variants in the general population. So if you use EXAC or Nomad, you can look at, let's say, minor allele frequency less than 0.1% variance, and compare the observations that you have in the general population to the expectations from your model. So this is a little bit unfair, I should say. Just I'm big on citing limitations. The Nomad model uses synonymous variants in well-covered regions to train up, it's actually a regression, to train a regression to go from the probability of mutation to the expected number. Then we apply it to everything. So what we're looking at on the x-axis is the expected number of rare variants coming from this mutational model, and on the y is the observed. And so when we look at synonymous variants, thankfully, potentially unsurprisingly, they fall right along that observed expected line. So that's an R of 0.97 or 98. So that means per gene, we're extremely good at predicting the number of rare synonymous variants. So given that, we trust that we can predict also the number of rare missense mutations and the rare peel-off mutations for all of these genes. The missense you still see for the majority of them, they're falling on this observed expected line. You have a couple points up here, but the correlation is still really high. However, as we move to loss of function, you can really see this drop off the observed expected line. So again, our model is selection neutral. It's just saying based on the stretch of the sequence, the methylation information, and how well you've covered it, this is how many loss of function mutations you should see in the gene. And we should potentially be unsurprised that essentially every gene is depleted for these expected loss of functions. So we are viewing that as a signal of the selection itself. Clearly, loss of function mutations as a class are generally considered damaging, right? They take out a copy of a gene. We think we have most of our genes for a reason. So a lot of the work over the last couple of years has been specifically trying to quantify this depletion to try to get a sense of, can we identify particular genes that are sensitive to the loss of a single copy? So here we're going to shift into talking about how we identify these constrained genes, but I'm happy to take questions while I drink coffee.
Sorry, there was one question online which had asked specifically about the hotspot mutation sites you'd identified. Does age impact with like the ability to detect those hotspots, and are they kind of clustered around telomeres at all?
Right. So I believe this is asking about the clustered maternally inherited mutations. And to answer the second part first, yes, a lot of the clustered of maternally inherited mutations tends to be telomeric. So it's on the very ends of those. Distance to telomere and distance to centromere is something that was included in another recent mutational model from Nomad to look at non-coding mutations. So we do think that that influences it. In terms of other hotspots, I did elide over it, but de novo mutations have a well-established relationship with age. So the older the parents are, the higher the de novo mutation rate. This is more noticeable for fathers than for mothers. Again, sperm are kind of constantly passaging and accumulating mutations across a lifetime. And there have been a bunch of estimates from Iceland, from studies in Utah, from studies in the UK, to try from the Netherlands, to try to estimate exactly how many more mutations are gained per year, but it's also family specific. So any of these high level estimates are kind of collapsed across multiple families. So I actually don't know it off the top of my head, but it's a handful every couple of years. That's across the entire genome, where we expect to see about 40 to 100, let's say, across a lifetime. Great, let's get into constraint. Okay, the highest level oversimplification of why we think this is interesting is that if something's bad, you shouldn't see it in healthy people, right? Super massive oversimplification, I know that. So we're gonna get more into detail. So there's two major ways that this can show up. One, a shift in the allele frequency spectrum, meaning that if it's bad, it should be kept rare. We shouldn't see it common in the population. And the second is fewer rare variants than we anticipate. So it's so bad that we don't even see rare variation in this particular area. So for focusing on the first one, this shift in allele frequency spectrum, kind of back in the day when we were first doing exec, there was this idea that the proportion of singletons should be a good indicator for how deleterious a class of variants was, right? So for example, loss of function variants should be more singletons in the data set. They should be more rare. But this has an underlying assumption about the fact that all mutational classes are equally mutable. So these are the results we saw initially from exec for proportion of singletons. So right, essential splice and frame shift are way up here in the highest proportion of singletons in exec. But then we saw the next one, instead of stop gained, adding a nonsense mutation, it was stop lost. So that can be deleterious, right? A stop codon's missing, but it shouldn't be as bad as adding a premature stop codon really early. Like that was kind of a surprise. We also saw that in-frame indels were apparently not quite as bad as like 5' UTR or extended splice or stop retained mutations. What is happening? Well, it's because, oh yeah, great, I highlighted some, too late. It's because some contexts are more mutable than others. Our good friends CPGs are back here causing problems. And in particular, there's certain mutations that don't have as many CPGs contributing as others. So we did see a strong negative correlation between mutability and the proportion of singletons for synonymous variants themselves. So this is indicating that actually mutation rate should be taken into account. So these contexts aren't equally distributed, and stop loss variants can't be created, for example, with a highly mutable context. So that proportion of singletons is inflated because contexts with CPGs were being recurrently mutated and we were seeing them more often. So we came up with MAPS, or the mutability adjusted proportion of singletons. This is that same score, just adjusted for the fact that we know some contexts are more mutable than others. And now it looks kind of sensible. We've continued to use MAPS for a number of other things. So obviously MAPS on mutations where we have some sense of what's important or not, we're just sort of reinforcing the knowledge we had. We have also applied it to 5' UTR variants. And here I'm showing you extended splice site variants. So the kind of classical splice site is the two bases right adjacent to the exon. And we can show that if you look, for example, at the donor +5, they have a really high MAP score as well as this kind of donor -1 site. And this is kind of putting it above the line of missense mutations in terms of MAP scores in exec. So this is another way that we can highlight it. So MAPS works on mutational variant classes, but can it work for genes? And the answer is yes. So this is work by Slavoj Petrovsky and David Goldstein, actually one of the earliest constraint scores out there. And it was looking at the balance of common functional variants, so missense and peel-off variants, to all variants in the gene. And really the idea here behind this residual variance and tolerance score is that you can identify genes that don't have as many common functional variants as you'd expect, given all of the variants that you see in the gene. So those in red were the ones that were highlighted as being depleted of these common functional variants and therefore intolerant of mutations. So this is one way you can do that. We're going to focus again a little bit more on fewer variants than you'd anticipate. And that's because, again, this is the model I've been setting up this whole time. So the idea, right, DNA is going to mutate. And in a gene that can tolerate mutational change, you'll see that, for example, things are lost in even tolerant genes. The constrained genes, that are intolerant to mutation, a lot of the mutations that arise lead to a negative outcome, and so they are removed from the population. So what you see over time is this depletion of the expected amount of variation, and that expectation, I will verbally and physically underline with my hands, it's coming from our mutational model. So if we expect to see 100 loss of function variants and we only see 10, that's probably, we're guessing, because of constraint, as long as we've corrected for other stuff. So we've tried to quantify this a couple of ways. The first, the oldest, or one of the oldest, is PLI, or the probability of being loss of function intolerant. And the idea here was that there's kind of three broad classes of genes in terms of their tolerance to loss of function mutations. Let's say tolerant, olfactory receptors, recessive, that are tolerant to loss of a single copy but not both, and then haploinsufficient. And really PLI with the probability of falling into that last class based on the depletion we saw in known haploinsufficient genes. So as a class, for example, haploinsufficient genes had 9% of their expected loss of function variation, where curated recessive genes were up at 46%. So these are different signatures here. And we were able to show in this older paper that known haploinsufficient genes, those essential in cell culture and that were dominant, were more likely to be in this most haploinsufficient, or this high probability of being loss of function intolerant tail. So this is a very bimodal distribution. We set 0.9 or above as kind of a, if you want a dichotomous cutoff line, that's where it was. But you'll see that recessive genes are depleted here. And again, doesn't mean they're not important for the disease. It just means they don't look as depleted as these haploinsufficient genes. But more recently, we've been trying to shift to a more interpretable and more quantitative metric, which is known as LUF, or the loss of function observed over expected upper bound fraction. For francophiles, it is supposed to be pronounced like the egg, and that was my best attempt. So PLI over here is this dichotomous metric. Again, most genes are kind of a yes or no, but we have a lot in the middle, and these were really usually small. For LUF, this is taking the observed over expected in our data set and putting a statistical bound on it, because we know that loss of functions are still rare. And even for the vast majority of genes in our data set, in 125,000 people, we still didn't expect to see that many loss of functions. So, 1 out of 10 is not as stable as 10 out of 100 or 100 out of 1,000, right? As the numbers get bigger, you're more confident in that estimate. So this is a statistical bound on it. And just to quickly highlight that it does perform as we'd anticipate. So haploinsufficient genes are in the lowest, most constrained LUF deciles. Autosomal recessive genes are spread across the tolerance spectrum. And olfactory receptors are in that more tolerant, less constrained metric. So LUF is what we have been suggesting moving towards, because again, it is a more quantitative metric, and it gives you a better sense of what this depletion actually means for PLI. So I'd be remiss if I didn't mention another whole class of constraint scores, the S-hat metrics. This is more in population genetic theory. So S-hat here is the measure of the selective effect of heterozygous P loss, so the loss of a single functional copy. And they can make estimates essentially from 0 all the way to 1. The closer to 1 it is, the more deleterious it is. And this is sort of the loss. You can think of it as vaguely in terms of this S metric is kind of the loss of reproductive ability in some ways, not in terms of like your ability to have children, but 0.1, for example, is you have 1 out of 10 less children than an organism has 1 out of 10 less children than you might expect. So these are a little bit more directly interpretable. Specifically, they're more interpretable than PLI, right? 0.45 to 0.9, as nicely pointed out in this other paper, doesn't mean anything, but an S of 0.1 to 0.2 is more directly interpretable. And I should say here, there was a recent paper or preprint from Jonathan Pritchard's lab that did a bunch of fancy machine learning, and we're specifically trying to apply this to smaller genes, because all of these prior estimates of S-hat or HS have been limited by what we see in the data set. So I should highlight here, though, no matter how complicated it is, it still has a mutational model in there. It still has that trinucleotide context and that CpG methylation. So all of these include this. At the highest level, I'm just going to highlight that these constraint metrics are really focused on really severely acting, more early onset. So if you're studying a condition that is more that is adult onset or more complex, these constraint scores might not pull it out. There's a population genetics theory that's really in detail in the Full et al. Nature Genetics paper. With that, just to say, missense is a lot more challenging. We've started to think about it, and there's a lot of different approaches here. This is just a high level to say that for missense variants themselves, instead of collapsing across a gene, we might actually want to look at, little domains within it, or across family members, or more recently, especially with AlphaFold, structural information. I will not go into detail here today, but I have a lot of the citations at the bottom of metrics that have started to look here. And then for non-coding variation, of course, we've started to look at this. So we created by we, I mean Conrad and Siwe, made a non-coding constraint model for the 76,000 genomes in Nomad v3, and have also been able to show for 1 kb windows, they can highlight 1 kb non-coding windows that seem to be more intolerant to mutations broadly. Non-coding is a challenge because we do not have that kind of knowledge of what's more likely to be important or not. We don't have the synonymous missense loss of function knowledge, and that does impact it here. But even with this constraint Z-score, they were able to pull out regions that, for example, were enriched for structural variants in developmental disorder cases compared to controls. So speaking of developmental disorders, this last section, which I will go through at the highest level, is about empowering gene discovery. So to take you back, like to 2010, exome sequencing is new on the market, and we were really excited as a field to be able to use exome sequencing to look at families with rare and severe disease, be able to tie de novo mutations in genes to those genes. So for example, Kabuki syndrome, they had 10 unrelated individuals, and they found that 8 out of the 10 in the initial cohort had a de novo loss of function variant in the exact same gene, right? So we have maybe 1 to 2, all of us have maybe 1 to 2 de novo coding mutations in our entire genome, and unrelated people all having it in the same gene. It's extremely unlikely to happen. However, the early ones, this was really, they were rare, they were severe, the thought that they were mostly monogenic or that these were major genetic contributors. When we tried to do this in autism spectrum disorders, we did not see something as obvious as that falling out. So our first paper looked at 175 trios, and we only had a handful of genes that had more than 1 de novo mutation, so definitely not this 8 out of 10 signal. And across the first 1,000 or so, we still only had 5 genes with 2 de novo loss of function variants. So for more complex conditions, this clearly, it was a little bit more challenging. But we hit this point where if you're going to be using multiple de novo mutations in the gene as evidence that that gene is contributing to disease, and we're sequencing a bunch of people and de novos just land, you better have a good idea of the expectation going into it. So this is where that mutational model, actually, this is what it was designed to do, to be able to say in a cohort of a given size, this is the number of loss of function mutations we expect to see. So we did apply it to, again, this is old work, but we applied it to about 1,000 individuals with autism. And we're able to say, yes, we see a significant burden of loss of functions across the genome, but specifically we have a handful of genes where we can say with a rigorous threshold that they are significantly burdened with loss of function mutations. Now, of course, this has expanded in the last couple of years. We have been able to, for example, take into account that not all mutations are created equal. For example, if you look at de novo missense mutations, the more deleterious they are by CAD scores, the higher the enrichment we see in our cohorts. And so we were able to create recently a mutation, a per variant weight based on, for example, a variant deleteriousness score, this SHAT score, and whether the missense variant fell in a region that was constrained specifically. And we're able to make this simulation-based framework. So for every single variant, it got a score, and then we ran about 1 billion simulations under this null mutational model, again, mutational models, to get a per gene enrichment score. And just to like move through this rapidly, this was able to identify close to 300 significantly burdened genes in 31,000 patients with developmental disorders. And some of the new ones, for example, are being driven by loss of function or by missense specifically, because we had missense as a separate test within that. And just to say that we know that there's more to come because the genes that we've associated have a higher loss of function mutability than some of the unassociated genes. So bringing it back, everything comes back. And these mutation rates are not just used in a de novo context. So the TDA method takes de novo and inherited information together. And this is part of it as well. So a recent autism study identified 72 genes. But again, the mutation rate was part of this Bayesian framework here. And so it's quite important to include. So we've talked a lot about mutational models and mutation rates and why they're important and cool, and how you can use these models to identify genes that are intolerant of damaging mutations in reference populations. So that helps not only for general knowledge. I mean, I think it's kind of cool. It highlights interesting biology, but it also helps us interpret rare variants and has aided in the ability to discover new genes associated with rare disorders. So it's not just the mutational model. We also know that genes that are intolerant to mutation are more likely to have de novo loss of functions in patients, for example, with severe developmental disorders. And those are my secret extra slides, apparently. Yes, this has like been a field's worth of work. I tried to cite everyone along the way, but I'm happy to send specific papers after the fact. Happy to take questions.
Thank you so much, Kaitlin. That was an amazing talk. There was one other question from the Q&A that was specifically talking about the PLOF models and how you incorporate short indels or frameshift variants into those models.
Oh my gosh, coming right for me. So these mutational models are great for single nucleotide changes, and no one has an indel mutational model that I think is reasonable. For these de novo studies, we have made hacks. I will call them hacks. My ex-advisors will potentially get annoyed at me, and that's fine. Now done it on something that's going on YouTube. So we sort of took the rate of frameshift analysis to get a sense of how many frameshifts we might expect in a gene. But indels, you need a larger sequence context included because a lot of them occur by replication slippage, right? If you have a really repetitive sequence and the replication slips. I've been trying to convince someone to do an indel model for like truly a decade. So if anyone wants a big problem in genomics, that's what it is. So yeah, these constraint metrics don't include indels at all. There are some more gene dosage sensitivity ones that include large structural variants. I didn't touch on those at all because they don't use that mutational model underlying it. It's more of an observed in cases versus controls going into that. And again, some fancy like ensemble learners in the back end. So we're hoping to get to a point where we can include them, but until we have a reliable indel mutational model, I'm not sure we're going to put them in our constraint scores, at least in NOMED.
Thank you.
Thank you for that talk, Kaitlin. You mentioned how the mutational model is trained on the non-coding genome, and with the recent work looking at differences in selection in the non-coding genome, I'm curious if you think there's indication to retrain in regions that don't have evidence of selection, or the empirical correlation of 0.97 that you see with synonymous variation is sufficient to say that it's doing all right.
So I think we've been happy with it doing all right, but we're finding recently that models that are a little off as we scale up become more off. So I think we're going to be reevaluating this quite a lot with V4 because we're jumping from 125,000 people being included to 730,000. So a minor difference is going to be a big issue. In particular, my team's already started looking into chromosome 16p because we actually, as Dan knows quite well, this part of the genome is a little complicated, and we do see that there's a clustering maybe of genes there that our mutational model isn't working well for at all. So this is where we need to move to something that includes better regional factors than what we're doing right now. And roulette seems like it's a little bit better behaved and might capture that. So to your point about taking non-coding regions that don't seem to be under constraint, we can certainly consider it, particularly as data sets get larger, genome data sets get larger. That gives us more observations to use. I mean, ideally, we'd potentially want to use de novos from healthy people, but except for decode, people don't really like sequencing full families just to have this mutation rate information, which is devastating to me. Almost all of the trios and de novos we have are coming from individuals with a patient has a severe disease, particularly a neurodevelopmental disorder. But as we sequence biobanks, maybe we'll be able to do this better. That's a good question, though. Good thing to think about.
Interest of time, to get ready for the panel, we have one more question from online. This is a broader question. A slide was shown of SNPs and how it differs across different populations. With pan-genome data out now and knowing how variations are ethnicity specific, how should we proceed or focus to get a better understanding of various disease?
So the pan-genome is a great advance here, and I think potentially a complication, and we haven't done a good dive into what the pan-genome fully means for our mutation rates. It's mostly that we want to be able to map there, but there's still not that much variation mapped to the pan-genome. And so, yeah, there are definitely going to be population-specific variations that haven't been measured before. And we are excited to actually explore that in the next couple of years. So the take home is watch this space. We don't have enough representation there yet to do much with it, but we're excited about it.